the recovery of linocellular waste. This research explores how to this abundant renewable source can be enhanced to produce biofuel, offering a cleaner and more sustainable energy solution for the future. The source of biomass, there are some lignocellulosic biomass. There are a lot of types of the source. We can find forestry residue, animal byproduct, industrial waste, organic waste, wastewater, and agriculture and core residents. In this work, used in the agricultural residents such as white brand and waste straw. In general, the composition and the structure of lignocellulose contain three component polymer. The first one is the cellulose. Cellulose is the polysaccharide contain the chain of the glucose linked with the tip of beta-1,4. The second polysaccharide is hemicellulose. Hemicellulose is the heteropolysaccharide contain the chain, lateral chain of the axilla, and different ramification for different type of the sugar. The last component is the lignin. Lignin is the type of phenylpropane derivative. So, for the hydrolysis and saccharification for this polymer necessitates a lot of type of the, or synergy of different type of the enzymes, as like cellulase. In the cellulase, we can find three type of the enzymes, endoglucanase, silibioderase, and beta-glucosidase. For the hydrolysis of the polymer xylan, we necessitate to find the enzyme endoxylanase and beta-xylosidase. For the lininase, generally, ce sont the peroxidase and the lacase. So, the objective of this work was to demonstrate how mix the two types of the strain of fungi, Trichoderma afroarzionum and Aspergillus niger, for to mix for these two both strain for the fermentation and production of the tip of the enzyme before to use it for saccharification. And for the biological material used in this work, two types of the strain, Aspergillus niger and Trichoderma afroarzionum. After sporulation, the spore was used for the fermentation for two substrates, white strand and white bread. Two types of the fermentation, solid state fermentation and submerged fermentation, was analyzed and used for to compare the quantity and of the production of the enzyme. Before this one, it's necessary to test it if there are the synergistic between two strands in the co-culture. And the test was realized in this figure, and we fermentation used just only with Aspergillus, the second with Trichoderma, and the third test with to mix the two strands. After fermentation, and extract was recovered, and the different type of the enzyme was analyzed, which endoglucanase and oxylanase activity, beta-glucosidase, beta-xylosidase, peroxidase, and lacase activity. Also, the protein of the concentration of the protein was measured with Bradford method. After testing the synergistic positive between two strands, the optimization of the production was used with two types of the optimization, as like OFAT, classical approach, one vector type, and RSM, response surface methodology optimization, using box, bank, and design as the matrix of the optimization. 4 parameters was analyzed: incubation time, moisture, incubation temperature, and the relation between two substrates, white round and white bread. In the condition optimal, the extract was used and utilized for the saccharification with pre-treatment and treatment substrate, and the hydrolysis, the enzymatic hydrolysis was realized at 50 degrees Celsius, and the reduced sugar was analyzed and measured via DNS method. The results of the reduced sugar was confirmed in the TLC. Here, the results for testing if there are synergistic between the co-culture Aspergillus and Trichoderma. We can say here the better activity was measured with the co-culture, and if you compare with submerged fermentation and solid-state fermentation, the activity is better with solid-state fermentation. And for this reason, continue the other parameter. For the optimization of the OFAT results was shown in this figure about for the different activity on the glucanase and hexaglucanase for the 4 parameter analysis. The first one for incubation time, the humidity, the temperature of incubation and duration of between the two substrate, and the optimum was fixed for the both activity as like endoglucanase and oxygenase. The result for RSM based in the box Benken design and the analysis of the variance was shown in this table for the both activity. The activity on the glucanase, CNCase and xylanase. The model is validated with the p-value registered lower than 0.05. Compared with Lacuvette, it's not significant. The model is valid, and we can start to study the different interaction and the effect of the different parameter. The effect of the linear parameter, some parameter is significant and the other no. The same for the result for the interaction between different parameters. So interaction is positive and significant, and the other no. And we can say here in the response surface plot is just showed the interaction between influencing sugar influencing factor, and the different here we can say the response and the interaction for both parameters, and we can say the remove and the variation of the response depend in the interval of the study. Before to test the saccharification, if you remember, the composition of the hemicellulose is heterosaccharide, and if you need to totally saccharified this one, necessitated a lot of type of the enzyme. This type in general is auxiliary enzyme, and tested different type of the enzyme is present in our extract route, and a lot of type of the enzyme present the activity. This is better for total saccharification of the biomass. For the result of the saccharification is shown in this figure, the figure A and the figure B present the comparison between treatment and treatment substrate. The first one is the result for the reduced sugar with treatment substrate, and the B present the treatment substrate with different variation of the reduced sugar. And in the parallel for each figure, we can show the if you want to compare for the quantity of the reduced sugar compared, it depends in the type of the substance. The first one, for example, the white brand and the Russian. You can say here the white brand presents the bitter and presents the high concentration and the quantity of the reduced sugar presented. This result was analyzed with the TLC, and the results of the figure of the reduced sugar confirmed in the TLC. The column, the A and the B, A present the untreatment reduced sugar and the B treatment sugar. For the first column is the standard. We can see the column, the name, the oligosaccharide used as the standard, the glucose and the oligo, donc hexane, C6. And the second column is the sample recovered with the white strand, 2, the white brand, and the third, the Russian. It's the same for treatment. By the consistent and the bond depend in the concentration of the dilution sugar. We can say here, for example, in the figure A and the column 2, it's very dense, and this confirm this result showed in the white bran, because in the white bran present a high concentration of the reduced sugar. It's the same, present the different type of the oligosaccharide. The purpose is the total saccharification. And the last one, I need to find the monosaccharides like glucose, arabinose, glycylose. And we can say the last bond, compared with the C1 is the glucose. It's very dense. This is the objective to produce the reduced sugar. So, just I finished for this conclusion. The mixed solid-state fermentation of Aspergillus niger and Trichoderma afroasianum successfully valorized white bran and straw into natural lignocellulosic biocatalysis. The co-culture exhibited strong enzymatic synergy, enhancing saccharification efficiency, particularly for a treatment substrate. This co-friendly approach offered a promising route for converting agricultural residue into fermented sugar, contributed to sustainable bioprocess and circular bioeconomy advancements. For this, I finished my intervention. Thank you for attention.